Good morning students, welcome to Nano 3 online classes. In the last video we discussed about translation. In that we have discussed what are the requirements in translation process. Okay, today we will see the mechanism of translation. So what is translation? It is a process of polymerization of amino acids from nucleotide sequence of mRNA ಒಳಗಿರುವಂತ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಸೀಕ್ವೆನ್ಸ್ ನಿಂದ ಅಮೈನೋ ಆಸಿಡ್ ಒಂದು ಚೈನ್ ಫಾರ್ಮ್ ಆಗೋದು ಈ ಪರ್ಟಿಕ್ಯುಲರ್ ಪ್ರೊಸೆಸ್ ಏನೈತಿ ಟ್ರಾನ್ಸ್ಲೇಷನ್ ಐತಿ. ಸೊ ಟ್ರಾನ್ಸ್ಲೇಷನ್ ಆಗ್ತೈತ್ರಿ ಸೈಟೋಪ್ಲಾಸಮ್ ಒಳಗ. ರಿಮೆಂಬರ್, ಸೊ ಇನ್ prokaryotes immediate after the translation process begins because there is no presence of introns, where in case of eukaryotes after transcription three modifications occurs, post transcriptional modifications. After that, when hnRNA becomes mRNA, this translation process in cytoplasm starts. Soon when mRNA releases from nucleoplasm. So what happens in this translation process? There are four steps. In the first step, there is an activation of amino acids. Second step is initiation. Third step is elongation, and the last step is a termination. So what happens in activation of amino acids? Soon when the modification takes place in mRNA, and when it leaves from nucleoplasm to cytoplasm, all the 20 amino acids they get activated. Yella 20 amino acids enagtave activate agtave because these amino acids are required in the next processes. So how they will activate these amino acids? They will attach to the particular enzyme. That enzyme is called amino acid tRNA synthetase. Amino acid tRNA synthetase. So these enzymes they are attached by amino acids. During this process ATP use ಆಗ್ತೈತಿ to phosphorylate, means two inorganic phosphates were released, and there is a formation of amino acid enzyme complex, and they contains adenosine monophosphate with them. Now the amino acids they are activated. So this is called activated amino acid complex. Activated amino acid complex anta ಕರೀತೀವಿ. Then these activated amino acid complex they move to their specific tRNA for attachment. This is anticodon loop, and this is amino acid binding site. Okay, so they will move and attaches here. As they will attach, in that process there is a release of adenosine monophosphate AMP and that enzyme. Okay, as they release these attachment of these enzymes, so you can take the example of this could be methionine, the first amino acid, then this is AUC. Okay, this is the anticodon of a amino acid. Now this is called amino acyl tRNA complex. Amino acyl tRNA complex. So this tRNA is a charge. Like that, all the 20 amino acids, they get charged, and all the tRNAs also they get a charge. So in the first step, all the 20 amino acids they got ready. They are attached to the particular enzymes, and that enzymes help that amino acid to attach the particular tRNA. So like that, all the tRNAs with their respective amino acids, they are ready for next further processes. Then the step second is the initiation. So what happens in an initiation process? Soon when mRNA releases from nucleoplasm and enters into the cytoplasm, the small subunit attaches the mRNA at the 5' cap end. Okay, and this attachment of small subunit to the cap region of mRNA, this is by initiator factor 3 in case of prokaryotes, and in case of eukaryotic initiation factor 3. So this small subunit attached to the mRNA at 5' end with the help of this factor. Now the first initiator tRNA which contains methionine, initiator codon, it comes and attaches to the region where its codons are present. So UAC is an anticodon. So UAC is an anticodon and AUG is a codon. Okay, so they will attach. So they attach at a P site. So the first site in a small subunit is a P site, and besides that there is a site, and A site is for the next particular amino acid. Okay, they contains a particular codon. So now they have attached to the P site. Okay, then the large subunit covers this entire area. The attachment of first tRNAs to the respective codons, it is by initiation factor 2, or in case of eukaryotes, it is eukaryotic initiation factor 2, the attachment of tRNAs. Next, the entire large subunit, when there is attachment of 2 tRNAs, it is called amino acyl tRNA complex, the attachment at P site and A site. This large subunit covers those tRNAs. Okay, so this large subunit attached to the particular mRNA and small subunit, it is by initiation factor 1 or eukaryotic initiation factor 1. Okay, then. So now this conjoined proteins, ribosomes, they will start the protein synthesis. ಏನಾಗೈತ್ರಿ protein synthesis start ಆಗ್ತೈತಿ. So this is about elongation, sorry, initiation. So initiation ಒಳಗೆ ಏನಾಗ್ತೈತಿ, small subunit mRNA ದ cap region ದಾಗ attach ಆಗ್ತೈತಿ, initiation factor 3 or eukaryotic initiation factor 3 help ಮಾಡ್ತೈತಿ attachment ka. Next first tRNA which has a codon of AUG methionine ತಗೊಂಡ್ ಬರ್ತೈತ್ರಿ. ಇದು ಅಟ್ಯಾಚ್ಮೆಂಟ್ tRNA ಗೆ ಪರ್ಟಿಕ್ಯುಲರ್ ಕೋಡಾನ್ ಅಟ್ಯಾಚ್ಮೆಂಟ್ EF2 ಏನ್ ಮಾಡ್ತೈತಿ ಯೂಕ್ಯಾರಿಯೋಟ್ ಒಳಗ, ಮತ್ತು ಈ ಇನಿಷಿಯೇಷನ್ ಫ್ಯಾಕ್ಟರ್ 2 ಪ್ರೊಕ್ಯಾರಿಯೋಟ್ಸ್ ಒಳಗೆ ಏನ್ ಮಾಡ್ತೈತಿ ಹೆಲ್ಪ್ ಮಾಡ್ತೈತಿ. ದೆನ್ ಲಾರ್ಜ್ ಸಬ್ಯೂನಿಟ್ ಏನ್ ಮಾಡ್ತೈತಿ ಎಂಟೈರ್ ಏರಿಯಾಗೆ ಏನ್ ಮಾಡ್ತೈತಿ ಕವರ್ ಮಾಡ್ತೈತಿ. ಸೊ ದಿಸ್ ಇಸ್ ಬೈ ಇನಿಷಿಯೇಷನ್ ಫ್ಯಾಕ್ಟರ್ 1 or eukaryotic initiation factor 1. ಇದರಿಂದ ಏನಾಗ್ತೈತಿ ಪರ್ಟಿಕ್ಯುಲರ್ ಹೆಲ್ಪ್. The next process is elongation. So elongation process ಒಳಗೆ ಏನಾಗ್ತೈತಿ, this large subunit and small subunit they will move from 5' to 3'. The mRNA will not slip. This entire ribosomes they will move in this direction. So that moving or that movement, the movement of ribosomes over mRNA from 5' to 3' is called translocation. ಇದಕ್ಕೆ ನಾವು translocation ಅಂತೀರಿ, and this can be initiated by elongation factors. ಇದರಿಂದ ಹೆಲ್ಪ್ ಆಗ್ತೈತ್ರಿ, elongation factors ಇಂದ ಹೆಲ್ಪ್ ಆಗ್ತೈತಿ. So this entire these ribosomes they will move over the mRNA from 5' to 3' direction. As a result, when they move, so these particularly AUGUU like that. Okay, so these codons which are present on mRNA they come out. So ಯಾವಾಗ ಟ್ರಾನ್ಸ್ಲೋಕೇಶನ್ ಆಗ್ತೈತಿ, ರೈಬೋಸೋಮ್ಸ್ ಏನಾಗ್ತಾವ mRNA ಮೇಲೆ ಏನಾಗ್ತೈತಿ ಫ್ಲೋಟ್ ಆಗ್ತೈತಿ, ಮೂವ್ ಆಗ್ತೈತಿ. So that time the first these units codons ಇವೇನಾಗ್ತಾವ ಹೊರಗೆ ಬರ್ತಾವ. As they come out, so this tRNA, this particular tRNA which is this methionine above this next amino acids, okay, then there is a formation of peptide bond. So peptide bond formation ಆಗ್ತೈತ್ರಿ, peptide bond formation dehydration process ಅಂತೀವಿ. Dehydration process it take place between carboxyl group and amino group. Okay, so like that, and this helped by peptidase. This initiated by the enzyme peptidyltransferase. Peptidyltransferase enzymes ಮುಖಾಂತರ ಏನಾಗ್ತೈತಿ ಇದು ವರ್ಕ್ ಆಗ್ತೈತಿ. There is a formation of a peptide bond between the amino acids like that. So when this amino acid is put on here, so this will separate it. When these codons are come out, codon ಹೆಂಗ್ ವರ್ಕ್ ಆಗ್ತಾವ ಹೊರಗೆ ಬರ್ತಾವ, ಅವಾಗ ಈ tRNA ಏನಾಗ್ತೈತಿ ಪರ್ಟಿಕ್ಯುಲರ್ಲಿ ಸೈಟೋಪ್ಲಾಸಂ ಒಳಗ ವಾಪಸ್ ಹೋಗ್ತೈತಿ. ನೆಕ್ಸ್ಟ್ this will shift to the tRNA which is present at A site, they will shift to P site, and next codon will enters inside this ribosome. So next codon ಏನಾಗ್ತೈತಿ enter ಆಗ್ತೈತಿ, then this tRNA will enters inside the ribosomes. Okay, so ಇದೇ ಟೈಪ್ ಏನಾಗ್ತೈತಿ elongation or translocation process ಒಳಗ continuously ಏನಾಗ್ತೈತಿ up to 3' and ribosomes ಏನಾಗ್ತಾವ ಮೂವ್ ಆಗ್ತಾವ. ಸೊ ಮೂವ್ ಆದ ತಕ್ಷಣ ಯಾವ ಕೋಡಾನ್ಸ್ ಔಟ್ಸೈಡ್ ಬರ್ತಾವ, tRNA ಏನ್ ಮಾಡ್ತಾವ ಅದರ tRNA ಔಟ್ಸೈಡ್ ಹೋಗ್ತೈತಿ, ಬಟ್ ಬಿಫೋರ್ ಗೋಯಿಂಗ್ they will attach their amino acids to the next tRNA or amino acid tRNA complex. Next this tRNA will take this place and the tRNA which is waiting outside, they will enter as their codons will enter inside the ribosome. So in particular ribosome so enter agate. So like that, there they will shift here, this tRNA will go outside, before leaving they will attach their amino acid. So like that, on one particular group of large subunit, the amino acid sequences are come out. So when they will reach to the terminal region, any one of these three UAA, UAG, UGA stop codons, any one of these stop codons, they are present at 3' end. As a result, this polymerization of amino acid stop, because they does not contains any amino acids. There is no amino acid for such a codons, so that there is no tRNA for such a codons. Okay, so these codons, actually they are associated with the releasing factors. They are associated with the releasing factors. Like releasing factor 1, they can associated with the UAG and UAA. Remember this. And releasing factor 2, it is associated with UAA and UGA. And one more, releasing factor 3, they will activate these two releasing factors. So totally 3 releasing factors irtave. Releasing factor 3 ಏನು ಮಾಡ್ತೈತೆ, releasing factor A ಮತ್ತು releasing factor 2 ಗ activate ಮಾಡ್ತೈತೆ. Releasing factor A, they are associated with these two stop codons, and releasing factor 2, they are associated with these two stop codons. Whenever these ribosomes they are nearby these any one of stop codon ಒಳಗೆ ಬಂದ ತಕ್ಷಣ ಏನಾಗ್ತೈತಿ, ರಿಲೀಸಿಂಗ್ ಫ್ಯಾಕ್ಟರ್ ಏನಾಗ್ತೈತಿ ಆ codon का ಬ್ಲಾಕ್ ಮಾಡ್ತೈತಿ. As a result of which the large subunit it is separated, small subunit will separated, mRNA will separated, and amino acid chain that is also get separated. So like that, this amino acid next really ಹೋಗ್ತೈತಂದ್ರ Golgi bodies further modification can okay. So like that, this particular translation process on cytoplasm olaga ಆಗ್ತೈತಿ. So there are thousands of such polymerization process take place in a cytoplasm. Okay, so this is about translation process, activation of amino acids, initiation, elongation, and termination. In the next, we will discuss about the expression of gene. Okay, that we will discuss in the next. Thank you.